WatBio is a collaborative project, and we really focused on using the very latest molecular genetic technologies to unravel some of the complex responses that plants have to drought. And the overall aim of this is to develop new germplasm that can grow and adapt to these very stressful conditions which are predicted over the next few decades. And the concept is that we'll be able to get more from less, so to grow these energy crops in a range of environments and thus make them much more sustainable. We're working on 3 species which have no role in the food chain. Poplar, which is a native European tree. We're working on miscanthus, which is a grass which doesn't occur in Europe but occurs extensively in Asia. And also arundo, which is a very fast-growing weedy plant that is all over the Mediterranean Europe. There are several values to working collaboratively in Europe, which are exemplified within WatBio. We're working across a very wide range of environmental conditions, so from northern temperate environments in Europe right through to the Mediterranean. And so working together enables us to consider all of those environments.
In Italy, we've planted 6,000 trees so that we can look at the drought tolerance in black poplar. And what we really want to do is find the genes that underlie the traits that we're really interested in, so drought tolerance. So what we want to be able to find is a tree that can maintain growth under water-limited conditions and conditions that are quite likely to be occurring kind of in the near future. So the reason that we have so many trees that we have to phenotype and look at is that what we're actually trying to do is find these genes that underlie drought tolerance in a really diverse germplasm base. So you have trees that come from across Europe, some of which come from a very arid region, some from much wetter regions. And what you can then find is the natural genetic diversity that we kind of harness in that. Gives you a lot of power to find the genes that underlie different drought adaptation strategies, different ways to tolerate the drought.
Association genetics uses the DNA sequence of lots of genotypes within a population to identify single nucleotide polymorphisms, SNPs, single base pair variations in the DNA genome, and then uses these along with the phenotyping data from the population to find statistical associations between particular genotypic markers, particular SNPs, and phenotypes of interest, so that we can narrow down regions of the genome that are important for a trait of interest like biomass yield or wood quality or drought tolerance. My PhD in association genetics in poplar is looking for associations between genes within the poplar genome and traits of interest, particularly drought tolerance, which means looking for traits that enable the poplar to remain high yielding under water stress conditions. And our hope is that these genes will be used for the breeding of poplar for a more sustainable, high yielding feedstock in the future in Europe. The main outcomes of my PhD should be a list of genes that are associated with things like biomass yield, with wood quality and with drought tolerance, so the ability to maintain yield under water-limited conditions. We are hoping that these will be used for the advanced molecular breeding of bioenergy poplar in Europe for its commercialisation as a source of cleaner, renewable energy for the continent. In addition to our Italian field site, we also have a site in the UK near Winchester in England. We've been using this site since 2009 to phenotype the black poplar population for biomass yield, for leaf development, for stomatal patterning and for wood quality.